So Monday will be the exam, and then Wednesday and Friday we'll have open sessions in here to study for the final exam. And so I'll give you some feedback on that as well, so that you have a guideline on what to focus on for the final exam. Questions on any of that? Next week, right? Lab presentations are not this Thursday, but next Thursday. That's why I didn't want to put the exam on the Friday, because you're busy doing the PowerPoints, and then to turn around and take an exam the next day would be challenging, I think. So I'm putting the exam first on Monday, and then you'll do the, so next week it'll be busy with that. Any other questions? Well, hopefully you had a little bit of chance to catch up on sleep and maybe have some fun over break. I know it's the big push time with regard to what's going on in classes, so just gotta push through it. All right, so right before break, we were getting into chilling and freezing ideas and that kind of stress that the plants are experiencing. And so I just have a few last things to say about that, and then we'll switch into drought and heat issues. So with chilling and freezing, I think we left off here, looking at the idea that you have species of plants that are more resistant to chilling and freezing tend to have a different ratio of fatty acids in their cell membrane. So they have adapted by actually configuring their cell membranes to have more of the unsaturated fatty acids. That's what you're seeing in this picture or table here. When you look at the very last row, it's showing the ratio of unsaturated to saturated. And so the higher the number means that you have more unsaturated. So you can see that the chilling resistant species have more unsaturated fatty acids in their cell membrane compared to the ones that are sensitive. And so plants have that ability to adapt they're pretty good at that phenotypic plasticity is the fancy way to say it. They have the ability to adapt to the conditions around them through their life. Now, chilling is one thing, freezing is another matter. And so when you look at plant tissues and you put them under freezing conditions, and they've done this for multiple different plants and different parts of the plant, and typically this is the pattern that you see, where you'll get ice forming more in the spaces in between cells. That's where it starts to first form. And then in the xylem vessels, those are usually the larger, the vessels are the larger diameter tracheids. Those cells, remember, they're all dead, so they're just kind of hollow tubes. You'll get ice forming in there as well. So the cytoplasm is kind of the last to go, which makes sense because once you have the cells and the cytoplasm freezing, then you got some serious problems. So what plants tend to do is try to create a situation where their cytoplasm is dehydrated. That will minimize freezing in the cytoplasm. And so that kind of forces the crystals to form more in the cell wall spaces in between the cells. And then in addition to that, plants will try to make specialized proteins, antifreeze proteins, so to speak, which there's other organisms besides plants that will do this. And so those are not like always there. They will only get made in response to really cold conditions, and they'll start synthesizing these antifreeze proteins with the goal of course, not having their cytoplasms freeze up. And so it does seem to slow down and stop the growth, so to speak, of the ice crystal. Crystals have a way of growing bigger and bigger and bigger over time and prolonged coldness. They will also ramp up the production of their sugars in their cytoplasm. And again, this is messing with how much free water is available in the cytoplasm. The less free water available, the less opportunity for ice crystal formation. And so if you can pack the cytoplasm full of sugars, and so there's usually straight ahead sugars, and then there's glycoproteins, which are sugars attached to protein that will get made in pretty high abundance as the plant is getting that signal, oh boy, I'm freezing, I'm under freezing condition. And so the plant is not totally at the mercy of freezing conditions. It does have some things to try to minimize the ice crystal formation, particularly in the cytoplasm. I want to try to minimize that. So these are just some of the things that they've noted. The fancy word for it is compatible solutes. You see that in the title up here, compatible solutes. So that's just an umbrella term that they give of different molecules that the plant can synthesize in order to essentially create an antifreeze-like condition in their cells. So I'm not going to get into the physics of it because it can get quite complicated, but there is this supercooling idea that plants can undergo, and it is a way to essentially prevent ice crystal formation in the cytoplasm. And so their ability to withstand the supercooling events that go on, again, that's getting into the physics of that can get quite complicated. But basically, the goal of the plant overall is to keep ice crystals small and keep them out of the cytoplasm. That's really what they're trying to do here. And so anything that they can do to create that situation, they will try, assuming that they are resistant to these levels of freezing. So when they look at the physics of it, they say about -40 C is the lowest temp before you get spontaneous ice formation. So like once you reach that temp, it's like it's going to happen no matter what. And so when they look at these plants as they're going through the process of cooling and then getting into freezing conditions, they do notice, in addition to the proteins and the sugars, they also see that abscisic acid hormone can get synthesized in response to the cold. And so when they noted that, they started looking into it a little bit more carefully. And, we already know that abscisic acid is used when the plant is sensing that the soil is drying out. The roots get that, oh, the soil is drying out. I need to think about ways of conserving the water I do have in my body. And so abscisic acid will be the signal that will get made in response to that idea that the soil is drying out, and the plant is definitely picking up on that. So when they looked at it with the freezing condition, what's going on is that if you take abscisic acid and you actually apply that to the plant, you actually get tolerance to freezing if you then put that plant under freezing conditions. And so what they see is the abscisic acid will get produced in response to cold, but you can actually manipulate the plant where you can, before it even has been exposed to cold, you can expose it to abscisic acid. And then if you do put it into the cold, it can handle it better. And so they're thinking about ways of, should we spray our plants with abscisic acid in the field, for example, if there's a freezing temps that are predicted for the weather report. This is an option that they're considering and probably have started implementing in some areas of the country where this is important toward the end of the growing season, where you have freezing conditions that come along even in August or September, especially around areas like this. And so the abscisic acid with this next slide here, and let me just also point out, with or without abscisic acid, like previous studies before they stumbled on the abscisic acid idea, you can get plants to acclimate to the cold just by short exposures to it and then bring them back in. So you do that every spring if you're a gardener. If you have tomato seedlings in your home waiting to be outplanted to your garden, it's usually you acclimate them where you put them out for a couple hours and then you keep extending that length of time that they're out so that they get used to the cooler temps in the spring compared to your house. And then you can also do that with abscisic acid. So what they're finding is that if you spray the plants with abscisic acid, that they can tolerate freezing conditions better. And what they tracked was that some of the same proteins that are made in response to cold, abscisic acid is doing the same thing. The same proteins are getting made in response to the abscisic acid that you spray onto the plants. And so they looked at, when they were doing this study, they took plants that don't pay attention to abscisic acid. They don't have the ability to hear it, so to speak, to respond to it, and they don't handle freezing conditions. And so they were just trying to have this kind of like a control for the experiment where they created these mutants. And they said, okay, what if the plant can't pay attention to abscisic acid? What do? And they don't do well with the freezing. They don't tolerate it. And then deficient mutants, what that means is that they can still pay attention to abscisic acid. They're not deaf to it, so to speak, but they're just not making it themselves. So then they created those as another type of control plant in this experiment where they said, okay, let's create the same plant that isn't making abscisic acid, but if you spray it, it does respond to it. And when they did that and had those plants, they could tolerate freezing as well. And so this experiment was basically just trying to set up the conditions to demonstrate that abscisic acid is necessary for these plants to withstand the freezing and be resistant to the freezing. So they're looking at that as a way to perhaps for growing plants in cooler environments, that would be a pretty easy thing that they could do if that would help the plants withstand freezing better. So it's not just the air temps, the freezing temps that the plant has to worry about in terms of freezing. They have long known that there are actually some bacteria that hang out on the surface of the plants that actually create microclimates on the leaf that trigger freezing conditions, even though the air temp is not quite at freezing temps yet. It's kind of weird, but these bacteria, they hang out on the leaf surface and they produce a very special protein that they excrete out, and it allows for ice crystal formation to form. They call it ice nucleation, and it starts to form around that protein right on the leaf surface. And what happens is that protein that the bacteria make and then the subsequent ice crystals that form around it, it's so significant amount of ice that forms that it will allow the tissue to freeze at warmer temps than what, like the air temp is not quite at freezing, and yet the leaves are freezing because of that protein that the pathogen is making. And so, of course, from the bacteria's perspective, it's a good strategy because essentially what it's doing is it's creating a situation where there's tissue damage. And then if there's tissue damage, that means the cells will break open and release all those yummy nutrients that the bacteria can then utilize. And so from the pathogen's perspective, it's a pretty nice way to get access to nutrients. Of course, if that is attacking your plants in the field, if these bacteria are present, even under cold conditions but not quite freezing, you can have freezing, quote unquote, freezing damage. And so when they discovered this, realized that it's actually the bacteria that are causing that problem, they actually came up with a pretty interesting experiment where they took that bacteria, it's a Pseudomonas species and of bacteria, and they created mutants of it that could no longer make that protein. And then what they did was they sprayed their plants in the field with all of that mutant bacteria. And essentially that kept the pathogen from gaining access to the leaves. And so just by spraying the mutant on the plant leaf surface, it kept the wild type normal pathogen from gaining a foothold and causing damage. So they were able to reduce the tissue damage quite a bit. Unfortunately, this was done in the early era of genetic modification, and it caused kind of a controversy because they were spraying out a genetically modified organism out into the environment. That caused some concern. So that actually was instrumental in helping kind of come up with guidelines for how to work with genetically modified organisms in research. You know, eventually typically you have to do field studies, but you have to do them under pretty controlled conditions. But anyways, it did solve that idea that you could kind of tip the balance in favor of the good bacteria and keep the pathogen at bay. Okay, I'm going to stop here with the chilling and freezing idea because I did want to introduce another stress that plants can get that is pretty important for many plants. Chilling and freezing is not as serious of an issue in terms of overall cost when you look at compared to drought. So drought is a big one. Look at that one next. Okay, so this map is showing drought-prone regions where if you see orange, well, the gray is desert. So there's like hardly anything going on there in terms of crop production. And then the orange, deep orange and kind of light orange are very high and high drought areas of the world. So if you just kind of look over this, you can see that there's quite a bit, especially when you factor in the gray, which is desert. You can see that there are large areas of land that are not really well suited for crop production. So that, of course, is a problem. The last couple plant biology conferences I went to, by far the largest symposium sessions were on drought and heat stress, and on all the research going into how do we deal with this. Especially the last one I went to right before COVID hit back in 2020. They were, excuse me, they were very concerned about the increase in the human population, and we don't have an increase in land mass to really sustain how many people are going to be on Earth, and once you get into the year 2050 and beyond. And so, if you talk to plant growers, people involved in crop production, it's a little concerning about what are the options and what are the difficulties. And just drought is a huge problem where you end up with huge crop losses, and it's so at the whim of Mother Nature. And I think this is one of many reasons why in the indoor agriculture business and that aspect of growing food has increased in interest. It's because you use far less water under those conditions compared to growing crops in fields. And so there's many options that they are looking at to try to deal with this idea, like what can we do? Of course, there's effort going into, can we grow plants in more drought prone areas successfully? And can we manipulate the plants to be able to withstand that? They're certainly looking at that. But they're also looking at, well, how else can we grow crops where we don't need to use so much water and we don't have to grow in desert-like environments. So here you can see again just the plant productivity, physiology, molecular responses, looking at the cost in US dollars, and you can see that drought and heat is by far the most costly stress that you have to deal with if you choose to grow crops in the field for a living. So usually they combine drought and heat because those usually go hand in hand. And so it's hard to really tease those two apart. So when you grow crops in a field of regular soil and you're looking at water stress and drought, definitely when is it hitting? When is that plant undergoing that stress? Is it right at the start when the seed is germinating? Is it at the seedling stage? Is it at the stage when it's shifting to flower production? A lot of effort is going into looking at what are the impacts during the different parts of the life cycle. And absolutely timing matters. They see big differences in responses by the plant when it undergoes drought and heat under different stages of its life. And, that makes sense. I think just from what little work you've done with the seeds in class from the start of the semester, if you don't keep that soil evenly moist when you plant that seed's going to die, so it's extremely sensitive to drought when you're looking at seed germination. However, when the plant is kind of in the middle of its life, not quite there yet for flower production, it's just doing its normal everyday living, drought, it's a little bit more resistant to drought under those conditions. So it definitely makes a difference. How long does the drought last? The heat stretch, how long does it last? Obviously, that'll make a difference in how intense is it. Is it incredibly hot versus just a little bit hot with a drought? And so you have lots of factors to consider. Here they're looking at squash. On the left here, figures A and B, where you have drought stressed plants in the lower image and compared to the top image. And oh my goodness, severe drought, those squash leaves are very, very unhappy. So we'll talk a little bit about that, the shift in morphology. And here on the right, you're looking at rice plants where it's not quite as dramatic in terms of they don't have broad leaves to show the dramatic shift, but you definitely see the overall downturn in overall growth for sure. So here, some data, corn and soybean, which are huge crops here in the United States. A little bit too big, but that's a whole nother story. So here you see periods of severe drought, and you can look at the yield, how it crashes. When you look at like the year right before it or the year right after it, you can see huge shifts in the yield, big impact. So obviously that matters to us because that's food that now we can't eat because it's gone. It's just the biomass isn't there. So they have lots of data to show that anytime there's severe drought, the overall yields are really impacted. Here you're looking at some plants that they created some mutants. So on the left is the sensitive one to drought, and this is Arabidopsis. Arabidopsis, if you recall, it's kind of like the guinea pig plant in plant physiology research. And so you can see the ones on the middle and the right one are mutants that are better able to withstand drought conditions. So clearly you can see that there's huge impacts on the plant with drought. And so now we're going back to the idea of water potential, how that when you have drought, you are definitely impacting the water potential of the plant. And so they do talk about a couple of different layers of drought. So they talk about water deficit, and that just means that kind of like the onset of drought, you're not quite in the middle of the drought yet. You're just getting into this. There's less and less water available. And they talk about ideas of how the plant might be able to postpone desiccation. They phrase it that way. So postponing it, they see that there's a certain shift in physiology where they are trying to postpone hopefully it's not inevitable, but often it is, the inevitable drought that's going to follow. And then they do find some plants have ways to tolerate it. They shift their physiology so that they can better tolerate the drought, at least for a period of time. And then if you do, these are under controlled experiments where they're usually tracking this very carefully. And then if you keep pushing the experiment where you keep denying the plant water, you start to see that the water potential of the plant body gets very negative, which means it's getting dehydrated. And so the leaves tend to, visually, you'll see the leaves starting to wilt. They don't stay fully open. They tend to droop or close up. And so that's a big deal because now you don't have the leaf blade at 90 degrees to the sunlight anymore. So that's going to impact your photosynthesis. And obviously, if your photosynthesis slows down, your biomass yield is going to start crashing. And so as the water continues to leave the plant body, you're going to get the cells actually getting dehydrated to the point where plasmolysis occurs and the cells shrink in size. The cell walls don't have enough water pressure pressing against them, so the cell walls even become less stable in their structure. Cell membrane tends to thicken up. You'll see that under the microscope. And you'll see that pretty much anything that relies on turgor or water pressure inside the plant, all those activities that are dependent on that will stop. And so a lot of what we're talking about here is the leaves, the leaf blade, instead of being nicely fully expanded out and relatively flat, it's going to be curled or drooped. It certainly won't grow in any sense. And then root cells also will be, even though they're not in the air directly exposed to the hot air, the soil, of course, can get really hot. In fact, sometimes the soil is warmer than what you see in the body of air above it because the soil is pigmented. And if you think about it, just like you wearing a black t-shirt versus a white t-shirt, you're going to feel that increase in heat with the pigmented soil. So sometimes the leaf or the roots can get hit very hard with the heat and drought, obviously. Here, there's just showing some data, looking at some key aspects of the plant body. So you can see the leaf expansion just tanks. So on the x-axis, it's the water potential idea, where you have, remember now, as the water potential gets more negative, that means the plant is getting dehydrated. And so you can see it doesn't take much for the leaf expansion to just plummet. And then you can see here, it's a little bit more gradual with photosynthesis. And so you can see that the plant is doing what it can to maintain its photosynthesis. But as you start to get into -1 onward, you definitely see a decrease. So now, if you're a plant, and especially if you think about some of the larger plants like trees or yeah, I guess a tree would be a good image to think about. Plants can decide, so to speak, that it might be a good option to amputate part of their body in response to the drought. And so plants in desert conditions, especially trees that are adapted, Sir David Attenborough has a good video of trees in the Zambia area of Africa and how they will just drop whole branches of themselves under drought conditions, even though the plant is still alive. And so, a lot of people think that the plant's dead. It's not necessarily dead. It might have just made the decision, so to speak, that it's better to get rid of part of my body than to totally die. And so this idea of going through abscission, where once the plant is assessing, the drought and heat conditions to the point of drastic measures are needed, they can self-amputate, whether it be some of the leaves that they'll decide to get rid of, or even whole branches will come off the tree. And so you've seen this figure before and looked at the hormone patterns that shift when you have leaf abscission going on, where if you have plenty of oxygen cytokinin in the leaf, that's telling the plant that the leaf is nice and healthy and keep it alive, everything's good. But as auxins and cytokinins decrease, that actually will trigger ethylene synthesis, and that's what's causing abscission to occur in the abscission zone of the petiole. So small decreases in the water content can certainly have a big impact on what's going on in the leaf and photosynthesis. But the plant may decide, I'll get rid of some of the leaves and hope that that will help me make it through this drought period and the heat period. So just because you see part of the plant has dropped off, the plant body doesn't mean that it's dead yet. There's still a chance. So it's actually quite a nice adaptation. Here again, you're just seeing leaf abscission in this case. So on the left, that one's watered well enough where you can see most of the leaves are on it. And then as you get into the middle and the right, you can see more and more water is being withheld, and you can see that there's more leaves missing on that core plant. So it's a good strategy. If you have to employ it, it'll at least keep the rest of the plant body alive for a while. These are some really aggressive experiments that were taken to the extreme. So you can see here the wild type plant on tobacco plants on the left. And if you've ever grown tobacco plants, the leaves are huge. They have beautiful broad leaves. And so you can barely tell that there's any leaves there. It just looks like terrible dead tissue. And so on the right, you can see tobacco that has been ramped up with cytokinin during drought. So even though the plant was experiencing drought stress, because of the extra cytokinin in the leaf, it was able to stay in it longer and not get the signal to die off. So cytokinin, as you think back to your hormone experiment, cytokinin retards senescence, it slows down. So that's why it's.
So those plants actually die first, though?
Well, so eventually, if you keep taking it to the extreme, like if you keep withholding, yeah, they are going to die because, yeah, I mean, you have all that leaf tissue to deal with during a drought. So, but at least they're interested in this idea with the idea of in the real world, you know, there are going to be plenty of drought stretches where there will be a rain event that could, you know, allow these to stay green, healthy long enough for the raining event. But yeah, if you kept these plants under drought stress, they would die too, for sure. Yeah. So they're hanging in a little bit longer than the wild type, but here again with corn, they have the so-called stay green variety that they sell. You can clearly see the difference here, the non-stay green variety. So they put that field under drought conditions. It says the stay green, during grain formation, they still had significant yield of grains. So that's what of course you're going for. You're looking for the corn on the cob. So again, it matters. When do you put that plant through the drought stress? When is it experiencing it? And obviously, as it's forming the corn on the cob, you don't want a drought. So from a plant's perspective, if there's, you know, in an environment where they're sensing, okay, I got less and less water in the soil, my roots are not feeling any moisture, it would make sense if you think about it from their perspective that they would shift their growth pattern to try to deal with this. And so the way to do that would be slow down your shoot growth and devote more energy into increasing your root growth to go search or forage for the water in the soil that currently, you know, around your current roots, there's nothing there. So go forage. And so they will see the shift where the shoots and leaves, that biomass slows way down, but you'll end up getting a lot more root biomass as they look for the the soil and the water. And then it makes sense that the plant would close down the stomata and try to hold on to any water that it has in its body before it's too late. And so of course that is going back again to abscisic acid, that you'll get the roots forming, synthesizing the abscisic acid and then sending it up to the leaves to tell the stomata to close down. So we talked about that when we got into the hormone section. And so that's why very often whenever you do plant physiology research, you always should do weight measurements of the roots separate from the shoots. You do dry mass measurements, but it's always looking at on the same plant what happened to the shoots and leaves versus the roots under the different treatments that you expose the plant to. So you've seen this already, just bringing that up again with the idea of when the drought time kicks in, you'll see the water potential start to get more negative. And then you'll see stomatal resistance increases. That just means the pore is getting smaller and smaller. So you get more frictional resistance. That's what they were referring to. And then you see this big upswing in the food again. And so you provide the water, and then you start to see the opposite. Everything starts to shift back into more normal state. Here, looking at abscisic acid, in again more directly with the wild type plants that has it versus the mutants they created that don't have any abscisic acid being made. And you can see again that the root to shoot ratio. So if you can see it's going up, that means there's more roots being, more energy being made toward root growth. That's why you see that red line going up like that. However, the blue line just staying stable. So they're saying that the root to shoot ratio really didn't. And so it's like the plant doesn't even really know that it's in a drought. It's kind of ignoring that idea. And so it didn't shift into trying to produce more roots. No, actually, well, it depends, I guess, on what you're thinking. But if you would want your plants in a field state, if they're growing in your field and they're going through a drought, you would want them to start kicking out some more roots so that they can get a little deeper, because hopefully there is some water down deeper they can access. So that's the thought with that you would want them. But there's a point, right? Because if you're trying to grow corn on the cob, you don't want a bunch of roots, you know what I mean? So it's like you have to be thinking about what the payoff is. And you have to be, it's getting better with technology. Like they have sensors now that they can bury in the soil essentially of the fields, and they can tell exactly what the water potential of the soil is. And so they can be much more precise on irrigation. Because there's a payoff, you do want the plant to not dehydrate, but you don't want so much energy going into roots that, because most of the crops are above ground crops, right? Like corn and cob. So you have to be careful about that. Usually they will irrigate under drought conditions, and they will try as well. And it really does depend on, like, is the plant about to make the corn on the cob? Then they definitely have to be very careful, making sure that there is drought going on. That will stunt the corn on the cob, for example. So there's a lot to it. There's a lot of data they have to collect. So a lot of farmers now, it's a lot of data driven decisions that they're making rather than just wandering the fields and picking up the soil and looking at it. They have sensors and computer systems that are monitoring everything. Here you're looking at the idea of dehydration, drought. So on the x-axis, you have the water potential in the leaf. So again, as it's getting more negative, that means it's experiencing more drought. And they're just showing two different responses. If the plants have acclimated to the drought, you can see that they are still able to carry out photosynthesis better compared to plants that weren't acclimated to that drought. And so there's a lot of research looking into that too, with if there's ways to better acclimate, get the plants used to it so that when they really do have to experience a true drought, they are better at dealing with it. And so here you can see it says in the legend, acclimated plants were subjected to a moderate drought for 2 weeks prior to the measurements. And so they were just kind of training those plants by a little bit of exposure to drought over time, and that got them acclimated. So plants are amazing in their ability to be very phenotypically plastic to their environment. They can shift quite readily. So here you can see 2 different things that they were looking at. They were looking at photosynthesis. So that's the red line. And then they were looking at what happens to sugar translocation. So what's going on in the phloem. And so you can see here that you have photosynthesis seems to be getting hit harder in terms of, as you see the water potential in the leaves get more and more negative, so more of a drought idea. You can see that photosynthesis is really impacted. However, translocation, it stays going better, a little bit better anyways, compared to what you see with photosynthesis. And so that was kind of a surprising finding. They were actually predicting the opposite. They thought photosynthesis would be the longer preserved process under drought, but it was actually translocation that did better. And so they kind of scratched their heads on that one and said, well, maybe that is allowing the plant the ability to get the sugar where it's needed for the best growth in order to deal with this drought. And so we're thinking about the idea again that when a plant experiences drought, it typically says put more energy into the roots. Let's get more root growth. Let's search and forage for water. And so they think that's why translocation was maintained better and longer compared to photosynthesis, because the plant was trying to get the energy reserves to the roots. And then also if the plant is starting to make seeds, that's the other idea too. When they do these experiments during seed formation, they could see that translocation is doing better. It persists through the drought better so that the seeds can get hopefully fully formed. Because obviously if you don't make your seeds, then you're not getting anything into the next generation. It's all for nothing. And so it does make sense when you look at it from this perspective that let's make sure that different parts of the plant body get the energy they need. So thinking about a typical plant that has its roots in the soil, it's you have that water potential in the soil, right? It's starting to become more negative. And then what happens, if the soil continues to dry out, you have that water potential in the soil getting more and more negative. And you can actually get where the soil is more negative water potential than what's in the plant's water potential. And so you can actually have water kind of being sucked out of the plant, going from less negative in the plant to more negative in the soil. And so if you keep the drought conditions severe enough where the soil gets that dry, you can actually see that situation. And so the plant tries to back off that, tries to minimize that from happening. And you tend to see big upswings in abscisic acid in the plant body. And again, you tend to see the root growth, the devotion to root growth over leaves or branches in order to try to get access to water. And so obviously the plant body doesn't want massive efflux of water out of its body. And so it will try to minimize that. And then one of the other things that it will be doing as it's along with abscisic acid and getting more root growth, it can try to flip the scale, so to speak, where if it can drive its own water potential to be more negative than the soil. And so again, if you play with the psi sub s of the water potential equation, so if the plant can get more solutes in its cells, then its overall water potential will get more negative, more and more solutes, more and more solutes, more negative, more negative. And so it can hopefully tip the balance so that its water potential in the body is more negative than what you see in the soil. And so they do form those compatible solutes that we talked about. And that is a way to essentially the plant is messing with the Sisebes of the water potential equation and making the Sisebes more negative and more negative with more. So yeah, it's a process where the plant has more than one way to try to deal with drought and heat. And again, these are just some ideas, sugars and amino acids, proteins that can get made. And it's just a way to try to mess with the water potential, essentially the Sisebes value. And so they have more than one way to try to deal with this, but it's still overall, it's a huge impact on crop production, drought and heat. And it's probably just going to get worse with global climate change. And on that note, that happy note, we'll stop there and we'll finish up a few more things on this idea on Wednesday.